Good morning, biology students. Hopefully you finished watching the video that I assigned over the last couple days here about the Human Genome Project. These were the two guys that ran the Human Genome Project. Francis Collins down there in the lower right, he's probably one of the smartest biologists on the planet today, if not ever. Really super amazing guy. He actually taught at U of M for a while, and he leads the World Health Organization now. Brilliant geneticist. He also writes really good books. If you're really into science and DNA and genetics and you're curious about like what's happening with the genetics revolution that's going on right now, like Francis Collins writes really cool books that are approachable by like normal humans. So I know you're in ninth grade and probably none of you just read books for fun, but I'm just telling you, he makes some good books. He has also got he writes about, he's very strongly religious. Not that I'm advocating any sort of religion or anything, but he has interesting thoughts on religion and science, which oftentimes in the United States we pretend like those two things are at odds with each other. But I thought it was important to point out that there's tons of religious people that do science for a living. There's no reason why those two things are at odds with each other. I always like to at least point that out. I'm not trying to advocate for any sort of religion or anything, but I think some people get this misinformation in their head that somehow science and religion are not compatible things. But anyway, if that's a topic that interests you, Francis Collins writes really good, thoughtful, interesting books that are not like above. A lot of times scientists have a hard time like talking to normal people, but Francis Collins does a nice job of writing a book that's readable. Whatever, you guys aren't going to read books anyway, but you could. I'm just saying, Francis Collins writes good books. Anyway, sorry, moving on. Hopefully you watch that video about the Human Genome Project and what the outcome is going to be of the Human Genome Project. At the end of that video, they went over some of the things that you could fix, even stuff like height and IQ of your kid. That stuff sounds scary. Like we're kind of heading into a scary world, and we're going to have to decide, like, I've said this many times before this year, but I don't think many people would have a problem with fixing kids with some horrible disease like cystic fibrosis or sickle cell anemia. Like, if we could fix those diseases, I think most people would say, like, yeah, we should do that. That would, like, lower human suffering. And, you know, these kids, people with diseases like that, with genetic inborn diseases like sickle cell anemia or cystic fibrosis or a whole list of them, multiple sclerosis. If we could fix those, we probably should. Lower human suffering, lower medical costs, because you'd actually like fix the problem rather than just treat problems throughout a patient's entire life. That would be a huge benefit to society. But then with the same technology, you could pick like how tall your kid was, or, you know, I want my kid to have blue eyes, so I'm going to genetically engineer them to have blue eyes. Like, you could do stuff like that. And that stuff gets into spooky territory with like, I don't know if we should be using that technology that way. So anyway, it's an interesting topic that your generation is going to have to deal with because this technology is happening now. U of M is one of the big leaders. I'm a MSU Spartan. I went to Michigan State for all my stuff, but U of M right now, U of M's medical school is one of the big players in genetics research. And I don't know, if you really are into this stuff, you think you might go into doing genetics type stuff for a career. You couldn't do better than U of M for a college choice. But anyway, just my 2 cents on all of that. It's important to understand the Human Genome Project because we're starting to get treatments and cures for horrible diseases right now out of the information that's coming from the Human Genome Project. Remember, the Human Genome Project was finished in 2003. They never would have gotten to the Human Genome Project had they not figured out the structure of DNA. So we're going to talk a little bit more about structure of DNA and what I want you to know about the structure of DNA. So let's take a look. Structure of DNA. DNA has a backbone and parts that look like steps on a ladder called base pairs. So remember, the whole thing ends up looking kind of like a ladder when we draw it all out, which we're going to do in a minute. And it's an important drawing, and I'm going to ask you lots of questions about it in the very near future here. So it looks like a ladder, and then that ladder has a twist. That twisted ladder shape, Watson and Crick coined that term. They decided that shape was going to be called the double helix. The backbone of DNA, it's like the straight up and down parts on a ladder. Those are the backbones of DNA. DNA has two backbones. They're made out of sugars called deoxyribose and phosphate groups, which we'll include in our drawing in a minute. So the backbone is just sugar, phosphate, sugar, phosphate, sugar, phosphate over and over and over again. And then the important parts, the code-carrying parts, are parts called base pairs. These base pairs are 4 molecules. These molecules are called adenine, thymine, cytosine, and guanine. And they're abbreviated ATCG because their chemical names are kind of long. Okay, but whenever you see somebody talking about DNA and they start talking about A's and T's and C's and G's, they're talking about these molecules that run across the middle of the molecule. These are the parts that look like the rungs on the ladder. They can only pair in a very specific way. Adenine can only pair with thymine, and cytosine can only pair with guanine. So the way I always remember that is that A's go with T's because it spells at, which is a word. Maybe that's not really the greatest way to think about it. And cytosines go with guanines because C's go with G's because a C and a G they look like the same letter except G's got a little widget on the end. So that's how I always remember that C's go with G's and A's go with T's. So let's draw our base pair diagram, or what our DNA is going to look like. And this is an important drawing, and I would make sure that I have a drawing like this in my notes if I were you. Let me switch over to a whiteboard here. And let's draw our DNA structure. We're going to start with the backbones. Actually, I lied. We're going to start with the key. So over here, I'm going to draw the key to my drawing. And our backbones are going to have 2 parts. They're going to have a stick-shaped part. That's going to be our phosphate group. And they're going to have a round part, because remember, sugar molecules have rings in them. So I like to make them represented with a round shape. Those are going to be, the round parts on my drawing are going to be my sugars. Okay, so now let's start my DNA drawing. Let's give it a title. This is going to be my DNA diagram, or my base pairing DNA diagram. And what we're going to have on this side is a sugar and a phosphate, a sugar, phosphate, sugar, phosphate, sugar, phosphate. All chemically bonded together. So these are all held together with covalent bonds, which, if you remember way back to when we did chemistry, covalent bonds are very strong. So the backbones of DNA, very strongly bonded together. The backbones are not going to fall apart. Sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate, sugar, phosphate. Okay, so we've got our 2 backbones of DNA made here. We're going to need to add some more parts. Okay, we've got our nitrogen bases. They're called nitrogen bases because these molecules contain some nitrogen atoms. It's just what makes them unique. So we're going to have 4 of them. We're going to have adenine, which I'm going to draw like that. Adenine goes with thiamine. Oops, forgot how to spell. I-N-E. And then we're going to have, that's fine, hold on. Here's a cytosine. And then finally, my last one is going to be guanine. Those are our 4 nitrogen bases. So I'm going to make up some genetic code here. Each one of these sugars would have a nitrogen base attached to it. So I'm going to make up some genetic code here. We're going to have an adenine here. And then let's do, I'm going to make a cytosine here. These can really occur in any order. Let's do another cytosine here. And then we'll do a thymine here. And we'll do a guanine here. Okay, so now I've got my nitrogen bases down one side of my DNA strand. So we've got adenine, cytosine, thymine, guanine, okay? These could occur in any order because this is really spelling out your genetic code. The order of these is readable. So remember that DNA code works a little bit like binary code in a computer. Computers have patterns of zeros and ones, and the computer can interpret those patterns and then produce images on your screen or make software run on your computer by interpreting the order of those zeros and ones. DNA works the same way. Your body can read the order of these A's, T's, C's, G's and make sense of the coded information that's in here. The information that's coded is giving your body instructions for how to build your proteins, and then proteins are the little molecular machines that go and do all of your chemistry that makes your body function. So this is spelling out the coding to make all of your body's chemistry work. Okay, so we haven't drawn the other side of my DNA yet, though. Talking about base pairing. So A's will only pair with T's. So across from that A, there would always be a T. T again is thymine. And I would repeat these words with me as I say them. Adenine, thymine, guanine, cytosine. You're going to need to know those words. So I would say them out loud. It helps your brain remember them. But look at how adenine, see how it's got this pointy shape, and then thymine's got this negative pointy shape that fits nicely. Now, these things are not really shaped like nice, neat little flags. They're actually molecules, but they have bonding opportunities that line up with each other. So adenine's bonding opportunities line up with thymine's bonding opportunities. And they make a hydrogen bond. They actually make a couple hydrogen bonds right here. And that's what sticks them together. Remember, hydrogen bonds are weak bonds. So this bonding opportunity, DNA can unzip down the middle just like a zipper. That's going to be important later on. The backbones don't fall apart because they're covalently bonded. But the bond between adenines and thymines, that's hydrogen bonding. Those can come apart and then rejoin, just like a zipper. Okay, so across from cytosine, what would we have? Cytosine, we'd get a guanine across from it. And that cytosine, we'd also get a guanine. Across from thymine, A's always go with T's, so it must be an A. A's go with T's. And G's go with C's. Oh, wait, there we go. See how easy it was to make that G into a C? All right, there we go. That's my base pair diagram. Now, the only thing wrong with this drawing is that I've just drawn a small, tiny little snippet of DNA. Actual DNA would go on for a long, long time in either direction. So that's why I'm drawing these arrows in here. DNA is billions of base pairs long. If I took the DNA in one of your cells and unraveled it and read out all the A's, T's, C's, and G's, I would have 6 billion pairs of bases. 6 billion with a B. That's crazy. DNA is a huge molecule, but it takes a lot of genetic coding, a lot of think of this as like your software programming in your cells. It takes a lot of A's, T's, C's, G's to spell out all the instructions for how to build your proteins and thus build you. So that's our basic structure of DNA. I would make sure I'm familiar with that structure of DNA. You should know about the sugar phosphate backbones and how the nitrogen bases pair up with each other. That's the structure. The other thing I've drawn wrong in my drawing, of course, is that we've just drawn it as a straight flat ladder. Now imagine taking that ladder shape and twisting it. DNA is this twisted ladder shape, and I'll show you some pictures of that in just a second. Okay. So there's one with a twist in it. See how the A's go with the T's and the C's go with the G's. So that's one pretty little picture of DNA. They didn't draw their sugar phosphate backbones very well. That's a more accurate model of what it looks like. Because remember, these are all atoms and molecules. So really, when you look at it, it just looks like a bunch of atoms and molecules stuck together. But you could see where the sugars are. If you look at this and remember back to when we did chemistry stuff. Look at the backbone here. See how this has got a 1, 2, 3, 4, 5-sided ring shape? That's the sugar. That's the pentose sugar that makes up the backbone. And this stuff that's holding together the sugars, those are our phosphate groups, right? And there's another sugar, and then another phosphate group, and then this is another sugar. And see how we've got our bases are attached to the sugar parts? So here's our A's, T's, C's, G's in the middle. It's all the spherical things in the middle. All right. Oop, nothing wants to work now. I broke it. There we go. Okay. DNA is a self-replicating molecule, meaning that it can make copies of itself. It's one of the only self-replicating molecules that exists. It's kind of an amazing molecule. There are DNA nucleotides present in the cytoplasm. New strands of DNA can be constructed using the old strand of DNA for a template. And I just want to show you an example of this, and then later on we're going to get into how replication actually works. DNA replication is a good thing because remember, you made out of trillions of cells and you started as one cell big, but you had to make copies of your instructions and give it to every offspring cell since the time you were one cell big. So you've copied your original set of DNA over and over and over again, literally trillions of times. And you never make a mistake. You never screw it up. Even though DNA bases, there's billions of bases in each DNA strand. So trying to make copies of it, that's complicated business. But you never make a mistake doing it because it's a template-based system. So let me show you an example of what I mean by a template-based system. Hold on. DNA is going to make copies of itself by absorbing molecules from your cytoplasm. Cytoplasm, remember, is a mixture of molecules. It looks like the art there on the left. The molecules to build new DNA is going to come from the cytoplasm, but you're going to use an old strand of DNA as the instruction template for building the new strand. Okay, so your cells are going to gather up molecules from the cytoplasm and then use the old DNA as a template. Now, when I keep using this word template, anybody who's had any experience like sewing stuff would be familiar with sewing templates. So like the thing there on the left, that's a template for making a pair of shorts, okay? So you would like cut out those patterns and you'd copy them onto a piece of cloth, and then you could cut out the pattern on the cloth and then sew together shorts. But you could reuse the template over and over again. Just keep laying the same piece of paper on different pieces of cloth and you'd cut out the same shape pieces of shorts over and over again. And you could make millions of copies of the same pair of shorts by using one template. That's sort of how DNA copying works. It's a template-based system. So this diagram over here on the right is trying to show you how DNA would make a copy of itself. So here's our original DNA strand with the A's, T's, C's, G's running down the middle. What the DNA is going to do is unzip. One strand goes this way, one strand goes this way, and we're going to pull new nucleotides from that cytoplasm mixture that I showed you on the last slide. We'll pull these new nucleotides, new DNA building blocks, and stick them onto the exposed old strand. So each new resulting strand is half an old strand. And that means that we can't screw up the order of the A's, T's, C's, G's, because when we unzip the DNA and we expose this C here, for example, the only thing that could bond to that C is a G because their shapes fit. So only G would be in here, would be attracted like a magnet to that C. And across from this T, only an A would be attracted to that T. So we end up building two perfect copies of DNA from one original strand. So every time your cells go to divide, they copy their DNA by doing this trick, using an old strand of DNA as a template to make the new strand of DNA. And that way, both resulting offspring cells are going to end up with identical copies of DNA or an identical instruction book so that they run all their cellular biochemistry exactly the same way because they have exactly the same software instructions in their DNA. DNA is a self-replicating molecule. It makes copies of itself. All right. We're not going to get into steps of replication today. I just wanted to give you an overview of how replication works. So that's enough for today. Be aware that you have your LTA quiz tomorrow. It'll cover D1 and D3, which is what we've covered so far in this section of notes. So be aware that that's happening. I will give you some warning. I'm scheduled about far enough out to figure out where the unit test is, but not quite there yet. I think it'll happen by the end of next week that we'll have a unit test, but I don't want to set that in stone yet. But you definitely have an LTA quiz tomorrow over D1 and D3. If you have any questions, please feel free to email me anytime. I will talk to you later.